Okay, so these are the functional groups of the four main organic molecules, and there's a lot of stuff going on here. But first, we're just going to begin with carbohydrates, like most people begin with. And so the two main functional groups of any carbohydrate, whether it's a starch, whether it's cellulose, doesn't matter. The two main ones are carbonyl groups and hydroxyl groups. I don't know if you can see that here, but they're just carbonyl and hydroxyl. And so here you can see this is a glycerol aldehyde molecule. There's just a big name, but the point is here you can see OH, OH, OH again. And so all these OHs, all they mean is that they are a chemical functional group called hydroxyl. So hydroxyl just means OH. And then we also have another functional group called carbonyl, which I already said. And there's two different types of carbonyl groups. So the way that I think of the first one, aldehyde, which is right here, I think of it aldehyde end. So if you're talking about an aldehyde carbonyl functional group, it's going to be at the end. And so that's what differentiates this one and ketone, which is in the middle. So there's not really a cool thing I can think of for that one, but basically if it's not at the end, that means it's not aldehyde, and if it's in the middle, then it's definitely ketone. So those are like the differences. Here I kind of circled the whole entire thing, but carbonyl is just a C double bonded to an O. So here, technically this is just the carbonyl group, but I kind of circled the whole thing, because there's just hydrogens everywhere, so doesn't really matter. And then here, again, C double bonded to O. If two carbohydrate molecules, so two monosaccharides, synthesize via dehydration synthesis, they are going to be bonded by a glycosidic linkage. And that's just like an important term to note when you're taking the exam. Also, so now I think we're done with carbohydrates because you know the main stuff, that's fine. And then now we're on to lipids. So there's like three main types of lipids. So we have like the main one, triglycerides, which is basically just characterizing a lipid in its normal state. Then we also have something called a phospholipid, like the phospholipid bilayer in membranes of cells. And then we also have steroids, and not just the ones that people use to get all muscly. There's also like steroids meant for medicine purposes. Anyways, that's not what we're talking about here. But the functional groups of lipids, they vary a lot because of the how much they vary when it comes to real life. But normally, when we're talking about a regular lipid, it will consist of a glycerol molecule, and that's just a 3-carbon chain. So here, I don't know if you can see this, but there's an H and then a middle C and then an H and then an OH on the other side. And that's only one, but normally glycerol, I just wanted to get the gist of it, but glycerol is a 3-carbon molecule, so that would just happen, but with 3 carbons. And so it kind of makes sense if you think about triglyceride, because then there would just be 3 chains here. Then we only have 1 here. So yeah, and also because it's kinked, this is another thing to note. Since it's kinked, it will probably be a unsaturated fat, but this one's kind of weird because it has hydrogen bonds, which would make people think that it would be a saturated fat, because normally unsaturated fats are bonded with C double bonded to C, but that's a little bit too much. Anyways, so we have these, right? And the two main functional groups are carboxyl, and try to see the difference here. See how in carbonyl, C is double bonded to O, in carboxyl, C is double bonded to OH, and then single bonded to O. So that's an important thing to differentiate between the two. Carboxyl, COOH, whereas carbonyl is just C double bonded to O. So, but carbonyl is only in carbohydrates, it's not in lipids. Another thing is hydroxyl. So there's a bunch of hydroxyl groups here. You can see one in the glycerol molecule at OH that I underlined in red. And when these are synthesized, the glycerol and the fatty acid come together via dehydrogen synthesis again, or the condensation reaction, whichever one you want to call it. So water is removed, dehydration, and then they're synthesized, and what synthesizes them is called an ester linkage. So we have glycosidic for carbohydrates, and then ester linkage for lipids. And then we also have things called hydrocarbon chains, which is just a C and an H. So, like, for example, this would be a hydrocarbon chain. It's just a C and an H, but it's not really, like, hard to understand. Also, in phospholipids, say we just have one phospholipid here. So, yes, they have a glycerol molecule. They have two fatty acids, the two tails. And they also have a phosphate molecule. So that's just something important to note if you ever have like a graph like that on the mid-year, which you probably will. So they do have a phosphate molecule in the phospholipid. So that's pretty much it for lipids. I think I got everything. So now we're on to proteins. So proteins. The main structure of a protein is an amino group here, which I don't know if you can see very well. I'll try to move it as close as I can. But the amino group has an N, a nitrogen, and then it's bonded to either 3 H's, 2 H's. It really depends on the chemical state of the protein. But then you have the central carbon, and then you have usually an H above here, but that can vary too. And then I just made an R here in general, but that's an R. I don't know if you can see it, but an R group just means because it's not really specific, but that's where the variation occurs. So it could be a hydroxyl molecule. It could be a bunch of molecules all bonded together, but that's where they will be. And then we also have a carboxyl group. So amino, central C, R group, and carboxyl. Those are like the main ideas, I guess, of proteins. So carboxyl, once again, we have a C double bonded to... Oh, I did this wrong. That must be confusing for you. So C double bonded to OH, and then C bonded to O, just like we have it in lipids. If it was just C double bonded to O, then it might be confused with carbonyl, but it's not. It's C double bonded to OH. But when you're thinking about proteins, it's important to note that it's just amino and carboxyl. And so those are the main ones. Remember that. Amino, carboxyl. Amino has nitrogen in it. I made an important note here. You probably can't see it, but it says disulfide bridges, so that's what I'm going to talk about right now. So proteins have 4 main structural elements as they're going through protein synthesis. So I won't get into that, but I'll just get into their 4 structures. So as they're synthesized by the tRNA and they're brought over there by the mRNA and all that fun stuff, the primary structure is simply their amino acid sequence, just the raw sequence. And then the secondary structure, that's where it forms either a beta pleated sheet. So it looks like this kind of, and then there's like sheets in the middle like that. It's like diagonal, and then there's also an alpha helix. So it kind of looks like a DNA double helix, but I don't want you to get confused with that. But it's important to just visualize that as the alpha helix for the secondary structure. And then the third structure, or tertiary structure, is when all these other elements come into play with protein folding. So you have hydrophobic elements, hydrophilic elements, water-loving, water-fearing, or other way around, but you get it. And then ionic bonds. You have another thing called disulfide bridges. And so these things is just a sulfur molecule that's bonded to a hydrogen. So say that on one amino acid up here, there was a sulfur that had a charge, and then there was a hydrogen molecule down here on another amino acid that had a positive charge, and so these two would bond together, and that bonding would cause the protein to fold a little bit. So that's where the sulfur comes into play. So it's just important to know like the different elements within the proteins. So that would be one. And then so that's what disulfide bridges mean. It literally is making a bridge between the two amino acids. And the last structure, quaternary structure, is simply when two proteins, like two fully folded, they're all done, they come together. And so that is when, so it creates this crazy, like, just really big polymer, and it's just crazy. And so that is what that's what a protein one is. And also, the way that occurs, the quaternary structure, is when a carboxyl group, or it's otherwise known as a C-terminal, bonds with another A-terminal of another amino group on another amino acid. And so that would be the N-terminal, I don't know why I said A, N-terminal. So C-terminal bonds to an N-terminal via dehydration synthesis, and so that synthesizing is called a peptide bond. So let's just quick recap. We have a glycosidic linkage, then we have ester linkage, and then peptide bond, or I guess you could say peptide linkage too, but that's like the main one. Alright, and so that's the main stuff about proteins, and then now we're going to move on to nucleic acids. So they store and transmit your hereditary information. The main structure, I didn't really draw a specific one, but DNA, RNA, deoxyribose, and then ribose nucleic acids, it all depends on the sugar right here. So the sugar is the main thing that determines whether it will be a deoxyribose or a ribose sugar. We have a phosphate group here. So that is one of the main elements of a nucleic acid. And then we also have a nitrogenous base. And so they're all kind of bonded. We have phosphate, sugar, nitrogenous base. And so what happens is when a nucleotide bonds or synthesizes with another nucleotide, that occurs through the phosphate group, because as you can see, there's an O-, there's charges on it, so it makes it reactive, and probably the most reactive portion of this entire thing. And then we also have sugars, which are mildly reactive, like the OH might have a charge or something like that. So those are the ones that could bond, but not in the same one. So say we have a phosphate molecule here, and then you have a sugar over here from another one, like another nucleotide I'm talking about. So then those two could bond, and so that synthesizing, that would create a polymer. But other than that, that synthesizing, that bond is called, this is a pretty long word, it's called a phosphodiester bond. Okay, so that's the bond that links one nucleotide to another nucleotide. I didn't draw that, but just imagine that it would be over here somewhere. Also, when we talk about nitrogenous bases, it really depends on the type of sugar you're talking about, but say we're just talking vaguely whether it's ribose or deoxyribose, we have two different types of nitrogenous bases. So we have purines, and so the purines are just A and G. And the important thing to remember about A and G, adenosine and guanine, is that they're both double ringed. So they have two rings together. One's probably bigger than the other, and they're just bonded together, like right here. And then also we have pyrimidines, and so pyrimidines bond with purines. Like that's kind of, it makes sense, right? DNA, you have A's bonded to T's and C's bonded to G's. Well, the A and G would be the purine in that, and then the pyrimidine, which I'm getting to in a second, is going to be the T and C, and also U if you're talking about ribose, and then the T would not be there. But for pyrimidines, they only have one ring. So there's only one for pyrimidines. And when I say pyrimidines, you probably can't see the letters here, but it's cytosine, tyrosine, and uracil. So those are the ones that are pyrimidines, one ring. And then purines, A and G, two rings. Another thing to note about nitrogenous bases, I promise I'm almost done. The C and G, they form a triple bond. So guanine and cytosine form a triple bond when they're bonded. And then A and T or A and U form a double bond. So obviously C and G are stronger bonds, so they will last longer if they're ever being tested, I guess. So these are the main ones. I'll go over their linkages first and the functional groups right now. So carbohydrates, we have carbonyl and hydroxyl, carbonyl, aldehyde, and or ketone in the middle. Then for lipids, we have carboxyl right here, and that will be on the fatty acid chain. We also have hydrocarbons on the fatty acid chain, and then we also have a hydroxyl group on the glycerol, which is an alcohol molecule. And then, and lipids is going to be an ester linkage, carbohydrates, glycosidic linkage, proteins is peptide bond, and then nucleic acid is phosphodiester bond. And then once again, proteins, they have an amino group and a carboxyl group, but don't forget about the disulfide bridges in the third structure when the protein folds. And then nucleic acids, we have a phosphate group, a sugar, and a nitrogenous base. All right, that's all for now.